Okay, so here's a fairly familiar representation of ribosomes. Generically, because proteins that are present in prokaryotes and eukaryotes differ somewhat in their number and their size. But everybody has two subunits, one large and one small, and they're all made of a complex of proteins and RNAs. So the large subunit has 3 RNA molecules, and the small one has only one. And there's a large number of proteins, no matter whether you're prokaryote or eukaryote, at least 80 in both cases. So those two subunits will assemble and form the machine that will synthesize our proteins. So I think I probably said this, that the eukaryotic ones are larger and have more proteins than the prokaryote ones. Okay, it's a very complicated machine that catalyzes the formation of peptide bonds. And actually, the actual peptidyltransferase action, which is enzymatic, if you will, is actually catalyzed by the RNA, not the protein. So the ribosome is actually an example of a ribozyme, something we'll talk about a little more on some later date. Okay, so in terms of referring to the various ribosomal subunits, there's sort of an unusual historical nomenclature for that, and that is the unit being the S or Svedberg. So the small ribosome, the ribosomal particle in bacteria is 70S, and eukaryotic one is 80S. Okay, so these numbers are not additive. The larger numbers means they sediment faster and they're bigger, but they don't add. The small subunits of the prokaryotic ones are 30S and 50S. Well, that adds to 80, not 70. Same for the eukaryotic ones. So this is one of those historical things that won't go away, and you just have to get used to it. Okay, the same is true of the ribosomal RNAs, that they are named according to their sedimentation values. So for eukaryotes, the larger subunit, well, the smaller subunit has one RNA that's 18S, and then the large RNA for the large subunit is 28S. The large ribosomal subunit also has the 5S and the 5.8S. Okay, so that's just a matter of convention. That's how we describe them. Okay, it does take all 3 RNA polymerases to make a ribosome, and we'll hear a little bit about Pol 1 in a second. The Pol 2, of course, makes all the proteins. Pol 1 makes the ribosomal RNAs. So this is a polymerase that's totally dedicated to the creation of ribosomal RNAs for use in protein synthesis. The 5S is separate, and that's transcribed from its own separate gene by RNA polymerase 3. Okay, so one thing about the ribosomal RNAs, and I'll show the gene structure in a sec, they're processed out of a larger precursor. So Pol 1 transcribes them, and it's dedicated to that purpose. After that, they're extensively modified by the addition of methyl groups and pseudouridines, which are sequence-specific modifications. So it's a very complex process, starting off with the production of very large pre-RNA that gets processed in many ways to produce the final ribosomal RNAs that will function in translation. Okay, so the cell biology is kind of an integral part of this whole process because the ribosomes themselves are assembled in a subnuclear structure called the nucleolus. And the organization of this is very important to its function. Okay, first of all, there are a lot of copies of the genes that encode the ribosomal RNAs, hundreds of them, and they occur in tandem clusters in our genome. In us, in people, there are 5 actual ribosomal DNA clusters, which together will produce our ribosomal RNAs. Okay, now because they're clusters and the fact that there's extensive processing that has to happen, the nucleolus, which is where the ribosomes are assembled, is seen next to the NOR, or nucleolar organizer region of the chromosome. So what happens is that the ribosomal DNAs are there, all assembled together pretty much, and the nucleolar organizer forms the nucleolus surrounding those regions. So the nucleolus is actually physically surrounding the ribosomal DNA in our cells. Now, even though we have 5 clusters, we usually have only 1 or 2 nucleoli, because the clusters can share the same nucleolus. Okay, so the 45S is one big thing. It starts with a 5' external spacer and goes all the way through the cluster to include the end of the 3' external transcribed spacer. So it's a large RNA, which is ultimately going to encode the 3 final RNAs that are going to make up our ribosomes. So in order, we have our 5' external transcribed spacer, are 18S RNA rRNA region, an internal transcribed spacer 1, followed by the 5' S ribosomal RNA gene section, followed by the internal transcribed spacer 2, the 28S ribosomal RNA, and finally the external transcribed spacer. So this one large RNA is made that's ultimately going to produce all of those separate final ribosomal RNAs. Okay, so thinking about what the nucleus and the nucleolus look like, the nucleolus is a very distinct part of the nucleolus is, and it's not actually surrounded by a membrane, but it's still visible as a distinct structure within the nucleus. And that's where the ribosomal genes are located and where the ribosomes at least begin and complete largely their actual assembly with the proteins and with the RNAs, including all these modifications and cleavages that we'll see in a sec. So remember, nucleus has two membranes, an inner and outer membrane, and it also has nuclear pores through which macromolecules can move. Okay, so overall, the nucleoli are sites for transcription of rDNA, processing, modification, assembly of most of the ribosomal proteins, and the formations of various intermediate pre-ribosomal particles. Now, it doesn't get completely finished there. Some happens in the nucleoplasm, which is outside the nucleolus, but still in the nucleus. And also, some of it actually is completed in the cytosol after export. So it's a complicated process involving different components of the cell. So here's a picture of what they look like when they're almost done. We've got this very large particle made of several RNAs and a lot of different proteins, which are actually able to actually get out of the nucleus, even though they're so huge, into the cytosol, where the final maturation steps will happen. Okay, so the overall view of where we're going with this, we're going to talk a little bit about transcription of the rDNA gene and the 5S gene, processing, which is to say all the endonucleolytic cleavages which happen to the pre-RNA in order to find the final mature RNAs, extensive modification of the RNA transcripts, which involves a class of small nucleolar RNAs, Sno RNAs, which are actually guide the methylases and pseudouridine synthetases to the correct location on the ribosomal RNA. And then finally, assembly and export. This is a very long and complicated process. So here's another look at our ribosomal RNA cluster again. Okay, so there's actually hundreds of repeats. And a reminder, we transcribe it, the large thing, the 45S piece, to include the RNAs that will encode the 18S, 5.8, and 28S. And this is done by PAL1, which is dedicated to this process. So there are a lot of ribosomes. It is kind of shocking that there's a particular RNA polymerase that all it does is transcribe the ribosomal genes. But if you think about how many ribosomes there are and how important that is, it actually does make some sense. The important thing is it's transcribed as a long precursor RNA that must be processed. Now this is not splicing because we're not putting the pieces back together. We're simply cutting them up into smaller pieces. So it's considered processing, but it's not splicing because they're not getting ligated back. So I think I went through all this, all the different parts. Okay, what's odd about this, maybe not so odd, the endonuclease cleavages that are needed to chop out the final mature RNAs out of the precursor, those are done in a very orderly manner, and it produce certain intermediates that are then processed to their final forms. And we're going to show a simple version of this, and then a more complicated one that you don't need to know, which is then chopped up by endonuclease cleavages into the smaller parts that eventually become part of the ribosome. Now, the gruesome reality here is that it's actually quite a large number of specific cleavages, way in excess of the ones that are shown on the previous slide. So this is just to let you know it's actually much more complicated than it appears. But the ultimate end is that you produce an 18S ribosomal RNA in the cytoplasm, and then same for the 28S, you produce a mature form which eventually will function as a ribosome. So the next big thing about ribosomal RNAs is that they are modified extensively by the addition of 2'-O methyls to the sugars and the formation of pseudouridine within the molecules. So the mature ribosomal RNAs are extensively modified by literally hundreds of different modifications that are occurring at very specific sites under the guidance of the Sno RNAs, which we'll get to in a sec. I should point out that meanwhile, we have to make the 5S RNA. That comes from a different gene that's transcribed by Pol3, but the 5S makes its way to the nucleolus to assemble with the rest. Okay, now one thing to think about right now is even while the process is being made, while transcription is even occurring, you're going to be making and assembling the ribosomal process on the new ribosomal RNAs. And there's a lot of assembly factors also. So the guide-directed formation of the 2'-O-methyl sugars and the formation of pseudouridine, okay, that is done by small nucleolar RNAs that basically make specific base pairs between the guide and the target at specific locations in the pre-ribosomal RNA. So, of course, the methylation and pseudouridinylation reactions involve enzymes too, but the guide RNAs are what tell the enzyme what nucleotide or sugar should actually be modified. Okay, so here's a picture of a 28S RNA. You can see it has a lot of secondary structure for sure, where the RNA is actually base paired to itself or to each other. But for the point of this slide, the idea is whenever there's a little red dot or a little arrow, that's a place where the RNA has been modified, either by the addition of by formation of pseudouridine or by modification of the sugar. So this is very extensive, occurring over hundreds, literally, of sites within the ribosome. Here's another picture of the same thing, again showing the secondary structure, and the little green and red dots show positions where the ribosomal RNAs are modified. So a reminder of 2'-O-methyl, we've seen this before in a different context. This is a ribosugar within, for example, a ribosomal RNA, pre-ribosome. What we've done is we've taken the 2'O, OH, and modified it by the addition of a methyl group. So there's plenty of these in different settings. Okay, pseudouridine, however, is a little weird. That if we consider what uracil looks like, uracil is bound to the sugar by a carbon to nitrogen bond. The nitrogen is on the uracil that's bound to the sugar. Okay, in pseudouridine, the uracil has been basically flipped over. So now you have a carbon to carbon bond, no longer a carbon to nitrogen bond. And the enzymes that are responsible are there in the nucleolus to carry these reactions out on very specific bases, nucleotides. Okay, so the Sno RNAs are small nucleolar RNAs, and their job is to guide the enzymes to the correct location. Okay, so needless to say, when you're doing a methylation reaction, you're going to be using an enzyme for that purpose, and the same for the pseudouridine. But they need to know where to go. So the way this works is that there are some small nucleolar RNAs which are actually, in part, complementary to specific target regions on the ribosomal RNA. So those guide sequences will carry the enzymes to the correct location to carry out the reaction. So these have kind of stupid names. One is called the CD box type, and that's for 2'-O-methyl. The word box just refers to the nucleotides that are characteristic of this type. And the HACA, or HACA type, is for formation of pseudouridine. But both types have their own weird base paired structures, as well as the guide region, which is going to tell it what nucleotides it should go to modify. So here's some snippet of where these things come from. They're actually found in the introns of protein coding genes. So they are actually, in some sense, transcribed by Pol2 as part of the protein coding gene, intron, etc. But they have to be processed out of the intron by specific nucleases, and then after that they can go to the nucleus, to the nucleolus, to carry out their functions. Okay, so here's one representation, not the usual one, of a CD type of Sno RNA. All right, so you can see that it's got a stem loop type internal base paired structure involving the 5' and 3' ends. There's also a region for which they get their name, which are called box C and box D. And there's actually 2 copies of those on this particular one. Importantly, there's a region called the guide sequence, which is generally 10 to 21 base pair nucleotides long. And that's the part which will actually base pair with the ribosomal pre-RNA in order to guide the methyltransferase to the correct location for methylation. So we've got this characteristic structure, some common sequences, but also the guides, which are going to be unique to the place being targeted, which is why, of course, there are hundreds of these to be able to make their way through all those target sites in the ribosomal RNA. Here's a more customary representation of these molecules, which are probably more like what they look like, but maybe not so easy to understand. Okay, so for box CD, we have the stem loop type structure, and in this case, we've got two target regions, which are complementary to the ribosomal RNA, and that's where the methylase will work. This particular Sno RNA has two different target sequences, and that's not unusual for these. Okay, the other type is the HACA type, and that's a little more complicated in its stem loop organization, but still you can see where the target RNA on the ribosome is base paired to a particular region of the Sno RNA, and that will guide this pseudouridine to the correct location on the ribosomal RNA. Okay, so here's yet another representation of the same thing. I think it shows a little better where the actual site of modification is going to be, though. The little stars indicate the nucleotide or sugar which is going to be modified. And we can see we've got the stem loop structures, internal base pairing, and target sequences, guide sequences, which are specific to whatever the location is going to be modified. Okay, so this to me is a little bit weird when you look at what's happening, the transcription and assembly of the early pre-ribosomes. Okay, this is a strange representation, but what we're showing is the ribosomal DNA. The little green dots are the Pol-1 RNA polymerase that's making its way along that template. So what's happening here is represented by the little blue and yellow and red dots, and what that means is that you're having proteins assembling there and modifications occurring even while the ribosomal RNA hasn't even finished transcription yet. So the pathway of ribosome assembly and modification starts even when the transcription has barely begun. And then when you get later, you're forming these various pre-ribosomal subunits that will eventually be matured to form the final ones. So this all is happening all at the same time in the nucleolus to form the pre-ribosomal subunits. Now the main one is this 90S pre-ribosomal subunit, which gets matured to form the pre-43S, pre-60S, and undergoes significantly more maturation before it eventually makes its way to the cytosol. Okay, so that previous slide's a little tough to kind of get your head around, but the idea, of course, is that this is an ongoing process, and processing, modification, assembly are all happening at the same time, even when the ribosomal RNA still hasn't been finished completely. So these are all co-transcriptional process. Okay, the important thing to remember, though, is that you have formation of these pre-ribosomal complexes that are eventually processed to form the final 40S and 60S ribosomal subunits that exist in the cytosol for translation. Okay, so again, another representation, we've seen this one before, is that the pre-ribosomal, almost done ribosomal subunits go from the nucleoplasm into the cytosol, where the further maturation takes place so that they can then participate in protein synthesis. Okay, important as this process is, it seems impossible that there would be mutations in it in the population which cause disease. But there are apparently some diseases which are associated with defects, or not complete defects, but perturbations in various parts of this process.